Uh, Thank you all for making the effort to stay till the last day. I hope that everyone is okay and well rested after yesterday. So today we have our session from genotype to phenotype, which is the fundamental question that we discussed also in the brainstorm session. And our first speaker is Tony Capra from UCSF, who will talk about algorithms for inferring phenotypes from ancient DNA.
All right, thank you. I'm Tony Capra from just across the bay at UCSF, and thanks to the organizers for the invitation to what's been a really enjoyable workshop so far. And thank you all for showing up, for the first talk here on the last day. So to motivate what I'm going to talk about here, what I want to talk about, this is not advancing. What I'm going to talk about here, I'm going to speak to that very fundamental question that I think all have touched on in some way or another in the work we do, and that's how does genetic variation influence phenotypic variation? And I think that most of us in our work, at least in some way, are thinking about this with the ultimate goal of understanding how genetic variation relates to variation in disease, or disease risk, or disease states, and better understanding of those states. So hopefully we can do something to better treat it. But when I reflected on the title of this workshop, from algorithms to discovery in genome-scale biology, I decided that it might be more interesting to talk about some work that my lab has been doing, thinking about a different opportunity I see for algorithms to really enable fundamental discovery in biology. And that's in using them to infer traits that are relevant to human evolution, in particular ancient traits. And this has really great transformative potential, I believe, because ancient phenotypes are really hard to obtain. We're sort of working from material that's coming from very harsh environments, like this Denisova cave in Siberia, that's been really a font of influential material in terms of our understanding of archaic hominins, or our close relatives. But what you get out is really decayed, limited material. And while I've been incredibly impressed by what the physical anthropologists can infer from a tiny bone fragment or a tooth about the individual that it came from, those data, they can't tell us about a lot of the traits we really care about. And I think there's great potential here because I'm sure, as most of you know, there's been a real revolution in the ability to extract and sequence DNA from ancient material like these bones and fossils. And so here's a map just showing locations from around the globe where ancient human DNA has been found and sequenced. And the end down there is 3,000, but really now it's more like 10,000 different individuals that have at least some genetic information, 10,000 ancient individuals that have at least some genetic information. These data over the last 10 years have been really transformative and revolutionary in our understanding of the recent history of our species. So in particular, they've revealed that recent human history was much more dynamic than previously appreciated, that migration, replacement, and admixture between populations were not exceptions, they were really the norm. They were incredibly common, going on all the time and throughout our history. And so as a result, modern populations we appreciate are very much combinations of different ancestral populations that are themselves the combinations of different ancestral populations to them. And these populations moved around quite a bit. And the available ancient DNA has really enabled these kinds of discoveries because you can infer these ancestral relationships pretty easily just from a sort of limited number of alleles. And so the methods that you need to sort of infer for those kind of relationships that are relevant to these questions are already pretty much there. However, I believe that the available ancient DNA has been way less useful, and it's been less revolutionary in telling us answers to this question. What were these ancient individuals like? What were their traits? How are they similar to and how are they different from those of modern human populations? And this question, I think, is particularly important when it comes to thinking about very ancient DNA, which, sort of arbitrarily I'll say is is DNA from individuals that lived more than 40,000 years ago. And there are now tens of such samples of humans and our closest relatives that are available, including a few high coverage genome sequences. And this I think is most relevant in terms of thinking about their phenotypes. One, because we know very, very little about them. And two, because they serve as a really useful close out group for comparisons to modern human populations here. So groups like the Neanderthals and Denisovans are our closest extinct relatives. It would be wonderful to be able to know a lot more about their phenotypes and how they interacted with their environments. And so these individuals branched off from the lineage that became modern humans roughly 700,000 years ago, and lived across a broad range in Eurasia for hundreds of thousands of years before modern humans ever even got into those environments. And as I said, we now have 4 high quality genome sequences from these ancient individuals from roughly 50,000 to about 120,000 years ago. And so to sort of restate this question, we're really interested in how do modern humans and their diversity relate to Neanderthals and Denisovans, these closely related groups. And I should say that the Denisovans, are particularly important when it comes to this idea of using genetic information to infer phenotypes, because they were not even known to exist before genetic information was able to be extracted from the fossils. The bone that sort of defined the Denisovans seemed like probably a Neanderthal, but until the DNA came out. And so, that reveals how very little we know about what they were like. So that's the big question. So if we're going to think about that, I think the natural thing to do is to go back to this context where so many of us are doing work and thinking, is that how do we predict phenotypes from genetic information in modern populations? And so I think many of you are probably familiar with, and perhaps this is the first place your head went, the concept of a polygenic risk score, or a PRS. And the basic idea of this is you go out and you find a population you're interested in, you genotype or sequence them, so you identify genetic variants across the population. Then you do a genome-wide association or study, or you look at an available genome-wide association study that's sort of estimated the effects of dosage of those alleles on the trait of interest or variation in the trait of interest. And then what's usually done is a sort of a simple linear model where the presence of the variance is weighted by the effects from the GWAS, and then the evaluation of that predicted risk or predicted trait value is associated with or you can be compared with the actual values, and then that can be applied to new individuals that you do not have the sort of phenotype information for, or you want to predict future phenotypes for. Okay, so I think an idea probably familiar to most of it, right? Unfortunately for this application, I think PRSs will not work for us. And there are a couple reasons I say that. So first, the PRS overall accuracy is not great for a lot of traits. So here's just one example from a paper from a couple of years ago that looked at a range of blood-related traits. And so on the x-axis are traits, on the y-axis is the R squared of the prediction to the observed trait in the UK Biobank European population. And so you can see that these are decent. There's definitely a signal there, but it's not great if you really care about the prediction for one individual. And so as a result, a lot of the excitement about PRSs these days is sort of looking at individuals at the extremes and sort of contrasting them and thinking about interventions that might work on them. Great for sort of clinical applications, but not necessarily for what we're trying to do here. You may also see that there are 2 different colors here. The red represents applying a PRS developed from a GWAS in European populations. The blue represents applying a PRS developed in the Biobank of Japan. So a population of East Asian ancestry, different from the UK Biobank population here. You can see these values are lower, right? So, and this unfortunately is a general pattern that's seen across a lot of polygenic risk scores, where you have decreases in prediction accuracy as you move further away in terms of ancestry from the population in which it was trained. Okay, and I think that makes sense when we think about the genetic architecture of these complex traits and the demographic history of human populations. You often end up with situations where the other population you might be applying it to will have variants that were just missing from the population that the GWAS performed in, and the varying effects for those that are shared may actually still be different. And so there's a lot of work going on in human genetics trying to improve the portability or generalizability of these models, but they, I think, are really not there for the idea that I'm proposing here of trying to apply it to an even further diverged population of ancient individuals. However, all hope, I believe, is not lost. There have also been some great advances over the last 5 to 10 years in machine learning methods for predicting molecular phenotypes from DNA sequence. So rather than trying to go all the way from genotype to organism level phenotype, they predict things like transcript levels or methylation levels or histone modifications, or as we talked about, things like AlphaFold that predict protein structure from sequence. And there are a whole range of such methods that are out there. And so my thought is perhaps we can use these sorts of methods to sort of get a more low-level molecular foothold on relevant phenotypes that can then be used to move up to these higher-level phenotypes that ultimately, I think, many of us are interested in. And so I'm going to give you one example of trying to do that today. And we'll focus on genome folding. And so I think if you were here a couple of days ago, this will be very familiar to you from John's talk. The DNA, our genome, is crammed into our nucleus. And the 3D structure of it, the relationships of where different parts of the genome are to others, is actually very relevant in influencing how genes are regulated and what parts of the genome may regulate other parts of the genome. So, for example, if you imagine this linear sequence here represents the DNA, and the colors are just indicating, so we can sort of indicate linear positions along it. There might be a region that's folded like this. There have been a bunch of assays that have been developed over the last 10 years to try to quantify this chromatin conformation. Hi-C perhaps is one you're most familiar with. What ultimately comes out is a triangle that looks like this, that relates the contact frequencies of different pairs of windows along the linear genome. So for example, in our little schematic here, the orange and blue bits are close together and thus much more likely to be nearby and in contact. And so we would have a red value here, meaning that these are close in 3D space, or much more likely to be in contact. Whereas the red and purple bits are quite far away in both linear and 3D space. So they get this blue value and we can kind of fill in the whole matrix. And what happens is you often see patterns that look like this, where there's contact domains or topologically associating domains that represent these kinds of loops where DNA within them is much more likely to be interacting with each other and regulating each other than even components that are closer by in linear space if they are outside of that tab. Okay, so this is, you're going to see a bunch of these. So that's how you interpret them. And in particular, my colleague at UCSF, Katie Pollard, published a deep learning model called Akita for trying to predict from DNA sequence alone, so basically a 1 megabase chunk of the genome, to predict the Hi-C map. So one of those maps, and this is just the same map, but symmetric, like I was showing before. And so what they do is basically, now a fairly standard sort of deep learning approach to predict at 2 KB resolution these contact maps, and then we can do that across the genome. And they find that these are actually, predictions are actually quite accurate, especially when it comes to the overall structure of these regions in terms of their 3D folding. Okay, so the idea is, can we now use methods like this to begin to think about this question? So I chose genome folding not randomly, but because we believe that, thanks to work of some people in this room even, that recent human evolution has mainly been driven by changes in gene regulation, right? So there are many ways that variants could affect this complex process of gene regulation. And so today we're going to focus on, the 3D structure. So basically trying to understand how the structure of the genome might relate to its expression and ultimate expression in phenotypes. And, of course, I don't want to belabor this point, but we won't ever be able to do Hi-C on Neanderthal material. All we get is basically, very, very small DNA fragments. We actually do have some ideas about how we might be able to try to reconstruct some contacts based on the sort of fragmentation patterns, but that's for another day. So the idea is, instead, can algorithms help us here? Can they help us infer what that 3D folding pattern might look like from a human sequence and a Neanderthal sequence, and then compare them? Okay. All right, so just the setup is basically, we have an input sequence from an ancient individual. We put it through this model, Akita, that was trained to predict 3D structures, and we get outputs that look like this. And we're going to ask a range of questions with this approach. So first, we'll just try to quantify, what did the Neanderthal 3D organization look like? As I said before, there are a handful of whole genome sequences of high coverage from 3 Neanderthals and 1 Denisovan from these locations here, here, and here. They cover sort of a wide range of geography and time. And we'll also apply these methods to a set of diverse modern human genomes from the 1,000 Genomes projects. So we get a quantification of kind of the diversity that's present in modern populations as well. We'll look at sliding 1 megabase windows along the genome, and then compare the resulting contact maps from the Neanderthal and the human contexts. And we expect that most regions of the genome will look like this little schematic, where they are very similar. We do not expect this to have evolved, substantially. But we also hope that we may find some regions that look like this, where there is divergence. And I'll say that the data I'll be showing you today will mainly be quantifying this divergence as basically 1 minus a Spearman correlation over the contact map. We've also done all of this looking at mean squared differences as well, and the patterns are similar. We also have a paper we're trying to sort of develop and benchmark other more biologically motivated comparison methods, but I've not applied them here. Okay, so the question is, what do these maps look like and how different are they? And so just to get your intuition going, I'm going to show a few examples. So here is a comparison for a given region on chromosome 16 of 1 megabase of the human contact map, the Neanderthal contact map, and then down here to help facilitate the comparison, because I know it can be kind of hard, especially on different screens, to detect these differences. I'm showing you essentially the delta in contact frequency, with green meaning more contacts in modern humans, purple meaning more contacts in the Neanderthal. And so here is a fairly representative region, very little difference. At the other extreme, here's a region that's at the 99.9th percentile of divergence according to our metric. And you can see, especially down here, there's this whole stripe of increased human contact, and then some focal bits here of increased Neanderthal contacts. So there's quite a bit of difference in the structure here that we think could be relevant. And then we see a full spectrum of differences sort of between those extremes. Oh, of course. Mm-hmm.
Assume that the sequence determinants are similar between.
Yeah, thank you for asking that. So as we saw from the polygenic risk scores, they did not generalize. I'm proposing that these kinds of molecular predictors are much more likely to generalize across populations and even species, because I don't think that the basic biology of how genome folding is determined by the sequence has dramatically changed. Now, we can't test that directly on Neanderthals, but we can test that by looking at chimp sequences. And I do have the slide showing, here we go. So here's an example comparing a human observed map and a chimpanzee, actually chimpanzee data on the top, where we see a very big difference between the human contact patterns and the chimp contact patterns for the corresponding region of the genome. And Akita captures this, at least qualitatively, very well. We see a decrease in performance across the genome in terms of the accuracy of if we apply the human model to the chimp sequence, in a context where we have actually an experimental chimp high-C map. It's maybe 10 to 20% less accurate than in the corresponding human region. So absolutely there is a decrease, but it's not, I think, as bad as in like the PRS case. It also depends a bit on cell type. And so that's another issue that I haven't directly talked about here, is that these models are based on 6 different cell types in a combined context. We also have cell type specific models. And in general, the sort of high level architecture of these 1 megabase regions is pretty conserved across the cell types. So we haven't delved super deep into that, but we have new methods we're developing that are trying to emphasize cell type specific differences that we are applying. So it's absolutely true that we don't think we have great cell type specificity with these models. In the chimp comparison, we've compared human models trained on different cell types, predicting the 1 chimp cell type we have data for, and they're pretty similar in how they generalize. Does that help? Okay. Yeah.
Question. So when you look at the gene expression, differences between species versus between organs, then you know that you have more similarity between species than between cell type. But that is a similar situation with the contact map, that they are more similar across the species if you use the same cell type, and there are more differences within the same organism.
Yeah, so it hasn't been anywhere near as well comprehensively characterized as gene expression across tissues and species is. The divergence in both dimensions, I think, is way lower for the 3D contact maps. But you would, I think, see, that's actually a good question. I don't know of a study that's had data across tissues from another species than humans. I mean, you can do it in mouse to human, I guess. There's enough data. So I don't know for sure, but I think it would be a lot more similar than.
More similar across the species than within the species and across the species. You don't know. Okay.
I don't know.
Just not that.
I think it would vary a bit across the genome as well.
I see.
Yeah.
Another follow-up question. It seems to me like in an anecdotal argument, why not train the program on chimp data or whatever mouse data?
Yeah,
Show us that
we have.
Okay.
I just don't have the plot here. And so that's basically where that like 10 to 20% reduction was coming from. If you train on human and apply across the whole chimp genome, the accuracy, decreases from like 0.2 to or I guess it depends on whether you do experiment or mean squared error. But yeah, it's about 15 to 20% of the error metric across the genome when you do that.
Yeah. So I'm trying to understand the biological basis behind the changes. And it seems that you need to account only for changes in target organization, which means you need to look only for mutations that affect the CTCF binding site. Otherwise, whatever outside of those regions, unlikely to change the organization.
So that's a very good point, that a lot of the signal that the deep learning method is finding is coming from CTCF, but it is by no means only from CTCF variation. So, and this is Katie's work, they showed that they get significant improvement by looking at the full sequence compared to like if you just quantified how many disruptions of CTCF motifs are there. So there is more on top of CTCF, but as I'll show you in a moment, often a lot of the drivers we think behind these differences are sort of characteristic changes in CTCF motifs. Does that make sense? Okay. So let me get back here. All right. So here is now the distribution of this 3D divergence score. In this case, it's like a 1- Spearman across the window, and you can see that most of the genome is very, very, very similar. And then there are a few out here that we're potentially more interested in. I think I'll skip this for time, but basically we showed that indeed there seems to be constraint on maintaining 3D genome contact. So if you simulate regions of the genome with similar divergence, similar mutation patterns, you would expect more difference than we actually observe between humans and Neanderthals. So the first question we have really though are there really Neanderthal-specific 3D contact patterns that were identifying? And so the way we we searched for this was to look for cases where all pairwise comparisons between a set of 20 representative African individuals were very similar to one another and small. And then the difference between the comparison of the archaic individual to all pairs of African individuals were all larger than any pair of African individuals. So we want to find situations where they are much, the Neanderthal to each modern human individual we have is much more different than any pair of modern humans, okay? And so that's in contrast to what most of the genome looks like, which is more like this, where the pairwise distributions of divergence are very overlapping. That means there hasn't been much difference. And so, we can sort of do some simulation to try to get a sense of the false discovery rate and put different thresholds on this. But at a low false discovery rate, we find about 150-ish regions. The exact number, of course, is contingent on whatever threshold you want to use. That we say have significant human-archaic 3D divergence. And here's sort of where they're distributed across the genome. And then here's how they are shared across the archaic individuals. And so, as you would expect from sequence patterns and evolutionary patterns, the Neanderthals are much more likely to share divergent regions with one another than with the Denisovan, who split off from their lineage not too long after they all split from the lineage that became us. And so this gives us a set of regions to start thinking about. And so, to answer to this sort of first question, yes, we can use this model. We can apply it to an archaic sequence and get predictions that essentially make sense given what we know and expect. So can these differences provide any clues to human Neanderthal phenotypic divergence? Just to give you one example, here's a particular contact map. Actually, in the interest of time, I'm gonna, I think, go through this a little bit quickly. So we can do examples, we can sort of do in silico mutagenesis using these models, evaluating different variants along the genome and find candidates that seem to be main drivers of the change. And as anticipated, in many cases, these are very interpretable given what we know about the biology of how these loops are maintained. And so, for example, here's a Neanderthal change that disrupts a CTCF motif, leading to increased 3D contact at the ICOS promoter. This is a cell surface receptor that's really important in cell-cell signaling and immune response. And so this sort of gives us a sort of way we could dig in and generate some specific hypotheses at this locus about differences between humans and Neanderthals. But we also, of course, can do the sort of things we all have to do when we want to generate larger hypotheses, and that's ask, are there any traits with genes enriched in the regions of 3D divergence? So imagine taking these regions, we tested for enrichment of annotations, both from rare disease, from the human phenotype ontology, and common disease from essentially the GWAS catalog, for genes that were in the TADs that are divergent between humans and Neanderthals. And for me, this was a really, I usually do these kinds of analyses and find some stuff that I can't interpret at all. This was really striking to me, what we ended up finding. So here is in the rare disease annotations, along the x-axis is basically just divided by disease domains. And this top dashed line is at a FDR of 0.05. The bottom one is at FDR of 0.1. We find some interesting traits, such as lower anterior hairline and underdeveloped supra-orbital ridges. And now, this is one of the most stereotyped differences between modern humans and Neanderthals, the classic strong brow ridge. So it was amazing to me to see something like this. And this is essentially saying that genes that are in these divergent 3D regions are enriched for being involved in the specification of a number of these hair and limb skeletal traits, okay? And so I think those genes now sort of become very, very interesting in terms of how they are regulated and how that might relate to some of these known differences. We also see, this set of sort of visual and optic phenotypes, which also actually correlates somewhat nicely with things that are already known in terms of Neanderthals having larger orbits and sort of differences in the visual processing machinery, at least estimated to have that based on the structure of their brain cases. On the, the common disease side, I think we could spend, hours or even days trying to interpret all of these. But again, there are quite a few, both, I think, reassuring observations here, and then also some provocative ones that we're interested in trying to follow up in more detail, specifically about those genes. All right, so of course.
All this is based on 43 regions.
No, well, we've done these for different subsets of these plots. I'm sorry, of these divergent regions. This one, I believe, is over all of them. Yes, this is over all 167. And the way we do the enrichment is by looking at genes that fall in the topologically associating domains. So it's not just that number of genes, it's a higher number of genes, a few hundred genes.
3 to 5 genes per count, something like that.
Yeah. And we do simulations to try to estimate what these FDRs really are, based on the fact that different genes have different probabilities of being put into a TAD based on the size of the TADs. So we sort of try to account for those biases along the genome in terms of gene size and TAD size.
So what is the, if you like compare, this is the divergence between Neanderthal and human.
And so it's basically, what if you look at other regions that have conserved TADs and the genes involved in them, but match to properties of the, the sort of size and gene density of the TADs that have changed, at least as well as we can through the permutations. Does that make sense?
But the cell type you use has nothing to do with some of these features you listed here. No, it's the lymphoplasma, right?
This is the combined model.
Oh, it's a combined model.
Yeah. And so I don't think we're getting a lot of cell type specificity. I think we're getting what are sort of like global, probably across most cell type changes between human and
And
Neanderthal.
within the TADs, you don't really care where the differences are relative to the gene. As long as they're within that TAD, you just
We've done it a couple ways. We've tried using just sort of nearest gene type strategies, and most of this looks the same. But yeah, in this analysis, we're basically looking just at genes in the TAD.
So sorry, since the two plots are different, so I take that you're only showing us the annotation specific for rare disease and for common disease, right? But then what happens if you just show the global map? I mean, how many things are not annotated there and have nothing to do with diseases or with traits that we cannot relate to differences between Neanderthal and us?
There are, if I understood correctly, we have very different power for different types of traits based on what we know about their relationship to disease in modern human populations. And that's in part why we've kept these separate, is they are just like different sets of mappings from genes to phenotypes. I don't have a great handle on.
So I guess the question is, do you find many things, terms that are enriched, but that we cannot really explain on or that apparently there's no difference between Neanderthal?
Yeah, I mean, like if we look
is this anecdotal or this is the norm?
Many of these are kind of on that spectrum. I mean, because a lot of these differences really are anecdotal. I mean, we are playing that game of, okay, what has been proposed based on the limited fossil information, or compare other comparisons of the genomes. And, qualitatively, many of these overlap well with those, as I was pointing out. Others, like, I don't know what to say about cannabis use, for example, in terms of how we interpret that. And I if anyone has ideas about solving this problem, I would love to talk about it, because I don't know how to sort of really get beyond the anecdote from these, other than digging into the specific genes and showing sort of how they were divergently regulated, showing that there was evolution, signatures of selection on those variants. That at least bolsters the case it's relevant, but it's still hard to necessarily say it was that phenotype. Yeah.
Into the Neanderthal and Denisovan introgressions, how much they contribute to the field organization.
I swear that I did not plant him in the audience, because that's exactly what we did next. So I think many of you likely know that modern humans and these archaic hominins overlapped in space and time at various sort of times and locations in our recent history. And, you know, somewhere around 40,000 years ago, there was interbreeding. And, you know, as I said before, this is sort of a common feature of human evolutionary history. People interbred when they could. And so there are lots of instances of this. This is inferred from sort of sequence information. But as a result of this, you know, somewhere around 40 or 50% of the Neanderthal genome remains, in a sense, in modern human genomes due to those admixture events. And so the question is essentially what I'm asking here. Did humans inherit any 3D organization from Neanderthals? I think that's the question. And so what we did is we took these 167 divergent 3D structures that we identified in the Neanderthals. And now remember, I only used African ancestry individuals in defining these because African ancestry individuals have very low levels of Neanderthal ancestry, just based on the fact that Neanderthals were outside of Europe, outside of Africa, and most of the interbreeding happened out there. There was a little bit of backflow, but the levels in African individuals is quite small. So our hope is that there might be signatures, well, I mean, yeah, I don't know if our hope is, but our idea is, can we detect signatures of any of these Neanderthal or archaic 3D contact patterns in modern Eurasian populations? And so just as an example of what I'm talking about, so here's a region, one of our divergent regions where there's an African contact pattern that is different from the Neanderthal contact pattern. So in the Africans, you have this sort of one large domain. Neanderthals, you have these two smaller domains, and there's sort of a CTCF motif here. If this, you know, so we then looked in the Eurasian populations at a range of individuals. Most people looked like this. They had the African, quote unquote, contact pattern, but there were several individuals that had predicted introgression at this location, and that resulted in the Neanderthal contact pattern. And so, this was really exciting. And it was not, and I have another example here showing that we can kind of show that introgressed allele is associated with gene expression, associated with phenotypes in modern human populations. In this case, it's a growth factor binding protein, and it's associated with height and some other BMI related traits. So again, something potentially evolutionarily relevant and relevant to some known differences between humans and Neanderthals, but also that may have been brought back to modern populations through introgression. Yeah. Yeah, we do have. So, and I should say that these examples are based on doing an in silico mutagenesis where we put in each variant in turn of the Neanderthal variants into the human background and scan those and find those that have the highest effect. But often, there are additional effects outside of it in terms of, it's not necessarily just the CTCF motif for sure. But we do have divergent examples where there is no obvious CTCF motif disruption, but those are by far the minority. Yeah. So I think you would get a lot of these if you just sort of looked at CTCF variants between humans and Neanderthals, but you certainly wouldn't get all of them. Yeah.
So those are for individual examples. When you look at the populations and the number of individual genomes that you have, does it pan out actually?
Yeah, so exactly. So the amount of introgression that you have in a 1 megabase window is really strongly correlated with how similar to the Neanderthal contact pattern you look. I have let me see if it's easy to get a quantification of that. Let's see somewhere. I'll come back to that.
Okay.
Thank you. Right at the end. I know I'm already a little over. Yeah. Okay. Okay. Sorry. So basically 24% of these archaic divergent regions, we were able to find in at least one modern human. And as I said, the amount of Neanderthal ancestry that an individual has in a region correlates with the similarity of their contact map to a Neanderthal. And so that is very significant. So in summary, I've sort of shown you how an example of how we can try to use sequence-based machine learning methods for predicting molecular phenotypes to hopefully understand better the molecular traits of long extinct or long gone individuals. And this is really just one example. I think there's great potential to be doing this with a lot of other traits. So we have a preprint out on this example, which I really encourage you to check out and send me any comments. I'd love to hear it. We're doing sort of experimental validation now of some of the introgressed human variants that I think will really sort of seal the deal. We have a paper from a couple of years ago where we did this on predicting transcript levels and similarly found some exciting differences. And that does get, that's a lot easier to get cell type specific given our better models for predicting cell type specificity of gene expression. And then we've got a review that we just published a couple months ago, summarizing all of the approaches in this field and pointing out that, we think this is a really promising way to go. So again, thanks. I'm sorry I went a little bit over, but I really appreciate all the comments and would love to talk more. Yeah.
One more question, which I think is kind of important. When you go back to the traits, there were some that we can easily see that there might have been selection for intelligence, and this is something that had Homo sapiens. But there were others that are more related to diseases, and I was wondering if you sometimes find disease traits in proximity, or genes for disease traits in proximity for beneficial traits.
Yeah.
And this could explain how beneficial traits dominated selection, and we are more susceptible to some diseases that the ancient populations might not have had.
Yeah, and I think you see this most strongly in the immune system, where an allele that may be detrimental in a modern environment might have actually been quite beneficial. So even maybe be the same effect can have a different, so the same variant can have a different effect on different environments.
Yeah.
Then of course also I think there is pleiotropy in many of these loci. And especially when you think about the 3D genome organization, there are multiple genes.
That's why that was really my question, that you have one gene that promotes intelligence, another one that predisposes to IBD, and the intelligence being selected for in the IBD kind of.
I think that's a good question. We haven't tried to use the tag information to look for potentially antagonistic interactions like that. And I think you could, that's good. I should. Okay, I'm good. Sorry, I apologize.
Sorry.
Yeah, thanks.
Thank you very much. Now we have Martin Fischer talking about predicting the effectiveness of the